So in tonight's lecture, we're going to continue with our discussion of cellular division or cellular reproduction. However, tonight the topic is meiosis. The first several slides are going to be review, so let's get to it. There are two kinds of cell division involved in the lives of sexually reproducing organisms: meiosis for reproduction and mitosis for growth and maintenance. So in order for us to understand both mitosis and meiosis, we must first discuss DNA, or deoxyribonucleic acid. DNA is the genetic material of living organisms. In humans, DNA is found in almost all cells of the body and provides the instructions that they need to grow, function, and respond to their environment. When a cell of the body divides, it will pass on a copy of its DNA to each of its daughter cells. DNA is also passed on at the level of organisms. When the DNA in a sperm cell meets the DNA of an egg cell, and they combine to form a new organism that has the genetic material from both parents. Physically speaking, DNA is a long string of paired chemical units that we call nucleotides that come in 4 different types. And it also carries information organized into units called genes. Genes typically provide the instructions for making proteins, which give cells and organisms their functional characteristics. In eukaryotic organisms, such as plants and animals, the great majority of DNA is found in the nucleus and is called nuclear DNA. In bacteria and other prokaryotes, most of the DNA is found in a central region called the nucleoid, which functions similarly to a nucleus but is not surrounded by a membrane. So remember that bacteria and other prokaryotic organisms lack membrane-bound organelles, which distinguish them from eukaryotic organisms. A cell's set of DNA is called its genome. Since all of the cells in an organism, with only a few exceptions, contain the same DNA, you can also say that an organism has its own genome, and since the members of a species typically have similar genomes, you can also describe the genome of a species. In general, when people refer to the human genome or any other eukaryotic genome, they mean the set of DNA found in the nucleus, that is, the nuclear genome. Moving on, each species has its own characteristic number of chromosomes. For instance, humans have 46 chromosomes, bees have 32 chromosomes, and dogs have 78 chromosomes. Like many species of animals and plants, humans are diploid, meaning that most of their chromosomes come in matched sets known as homologous pairs. Thus, the 46 chromosomes of a human cell are organized into 23 pairs, and the 2 members of each pair are said to be homologs of one another, with the exception of the X and Y chromosomes. The X and Y chromosomes are said to be hemizygous. In contrast, human sperm and egg cells, which have only 1 homologous chromosome from each pair, are said to be haploid. When a sperm and egg fuse, their genetic material combines to form one complete diploid set of chromosomes. So for each homologous pair of chromosomes in your genome, one of the homologs is maternally inherited and the other is paternally inherited. Moving on, so there are 23 pairs of chromosomes in humans for a total of 46 chromosomes in a normal somatic cell. And there are 2 types of chromosomes. There's autosomes. Autosomes are any chromosome that is not the sex chromosome, and the other type of chromosome is the sex chromosome, which is made up from either your X or your Y chromosomes. Females are going to have 2 X chromosomes, and males are going to have an X and a Y chromosome. And in the human, the haploid cells, which are your sperm and eggs, have only 23 chromosomes total. I believe that this haploid number in your germ cells contributes to sex drive, in the respect that since they have half the amount of DNA that a somatic cell would have, the sex cells or the germ cells need to find another cell to make themselves whole. It's a hypothesis of mine. This next slide shows what we call a karyotype. So the two chromosomes in a homologous pair are generally very similar to one another. They're the same size and shape and have the same pattern of light and dark bands. Bands appear when the chromosomes are stained with a dye, and the dark bands mark more compacted DNA, while the light bands mark less compacted DNA. Most importantly, the two homologs in a pair carry the same type of genetic information. For instance, there is a gene found near the bottom of chromosome 15 that affects eye color. A person might have the blue version or allele of this gene on one homolog, but the brown version on another. Both homologs have the same type of gene in the same place, but they can, and often do, have different versions of genes. Furthermore, in humans, the X and Y chromosomes determine a person's biological sex. As I've already stated, females will have two X chromosomes, and males will have an X and a Y chromosome. While the two chromosomes in a woman's cells are genuinely homologous, the X and Y chromosomes of a man's cells are not. As I stated earlier, we call that hemizygous. They differ in size and shape, with the X being much bigger than the Y. Actually, the Y chromosome is the smallest chromosome in the human genome. So we can say that women have more DNA than men do. Furthermore, the X and the Y chromosome contain mostly different genes. The X and the Y chromosomes are known as sex chromosomes, and the other 44 chromosomes are called autosomes. And that brings us to chromosomal structure and compaction. Each eukaryotic chromosome contains one very long DNA molecule, typically bearing thousands of genes. The number of chromosomes in a eukaryotic cell depends on the species. Chromosomes are made up of a material called chromatin, which is a combination of DNA and protein molecules. The protein molecules help to organize the chromatin and help control the activity of its genes. Most of the time, the chromosomes exist as a diffuse mass of fibers that are much longer than the nucleus they are stored in. In fact, if stretched out, the DNA in just one of your cells would be taller than you. As a cell prepares to divide, its chromatin fibers coil up, forming compact chromosomes. When they are in this state, chromosomes are clearly visible under a light microscope. When a cell is not dividing, the chromosomes are too thin to be seen under a light microscope. Such long molecules of DNA can fit into the tiny nucleus because within each chromosome, the DNA is packed into an elaborate, multi-level system of coiling and folding. A crucial aspect of DNA packaging is the association of DNA with small proteins called histones found only in eukaryotes. Bacteria do have similar proteins, but prokaryotes lack the degree of DNA packaging found in eukaryotes. So the figure on the left hand of the slide presents a simplified model for the main levels of DNA packaging. At the first level of packaging, the histones attach to the DNA. In electron micrographs, the combination of DNA and histones has the appearance of beads on a string. Each quote-unquote bead is called a nucleosome, and this consists of DNA wound around histone molecules. When not dividing, the DNA of active genes takes on this lightly packed, quote-unquote, beads on a string arrangement. When preparing to divide, chromosomes pack further. At the next level of packaging, the beaded string is wrapped into a tight helical fiber. This fiber coils further into a thick supercoil. Looping and folding can further compact the DNA, as you can see in the chromosome at the bottom of the figure. Viewed as a whole, this figure gives a sense of how successive levels of coiling and folding enable a huge amount of DNA to fit into a cell's tiny nucleus. Before a cell begins the division process, it duplicates all of its chromosomes. The DNA molecule of each chromosome is copied through the process of DNA replication, and new protein molecules attach as needed. The result is that each chromosome now consists of two copies called sister chromatids, which contain identical genes. The two sister chromatids are joined together tightly at a narrow waist called the centromere. When the cell divides, the sister chromatids of a duplicated chromosome separate from each other. Once separated from its sister, each chromatid is considered a full-fledged chromosome, and it is identical to the original chromosome. One of the new chromosomes goes to one daughter cell, and the other goes to the other daughter cell. In this way, each daughter cell receives a complete and identical set of chromosomes. A dividing human skin cell, for example, has 46 duplicated chromosomes, and each of the two daughter cells that result from it has 46 single chromosomes. And that brings us to meiosis, which is the basis of sexual reproduction. Only maple trees produce more maple trees, only goldfish make more goldfish, and only humans make more humans. These simple facts of life have been recognized for thousands of years and are reflected in the age-old saying, like begets like. But in a strict sense, like begets like only applies to asexual reproduction, because the offspring of asexual reproduction inherit all their DNA from a single parent. They are exact genetic replicas of that one parent and of each other, and their appearances are very similar. However, in a sexually reproducing species, like does not exactly beget like. You probably resemble your parents more closely than you resemble a stranger, but you do not look exactly like your parents or siblings. Each offspring of sexual reproduction inherits a unique combination of genes from its two parents, and this combined set of genes programs a unique combination of traits. As a result, sexual reproduction can produce tremendous variety among offspring. Sexual reproduction depends on the cellular processes of meiosis and fertilization. But before discussing these processes, we need to return to the chromosome and their role in the life cycles of sexually reproducing organisms. If we examine cells from different individuals of a single species, sticking to one gender for now, we find that they have the same number and types of chromosomes. Viewed with a microscope, your chromosomes would look just like those of Melania Trump if you're a woman, or Donald Trump if you're a man. In humans, a typical body cell called a somatic cell has 46 chromosomes. If we break open a human cell in metaphase of mitosis, stain the chromosomes with dyes, take a picture with a microscope, and arrange them in matching pairs, we produce a display called a karyotype. Notice that each chromosome is duplicated, consisting of two sister chromatids joined at the centromere. Notice also that every or almost every chromosome has a twin that resembles it in length and centromere position. The two chromosomes of such a matching pair are called homologous chromosomes. They carry genes controlling the same inherited characteristics. If a gene influencing eye color is located at a particular place on one chromosome, then the homologous chromosome has the same gene in the same location. However, the two homologous chromosomes may have different versions of the same gene. Let's restate this concept since it can be a little bit confusing. A pair of homologous chromosomes has two nearly identical chromosomes, each of which consists of two identical sister chromatids after chromosome duplication. Altogether, we humans have 23 pairs of homologous chromosomes. Other species have different numbers of chromosomes, but those two usually match in pairs. For a human female, The 46 chromosomes fall neatly into 23 homologous pairs, with the members of each pair essentially identical in appearance. For a male, however, the chromosomes in one pair do not look alike. The non-matching pair, only partly homologous, is the male sex chromosomes. Sex chromosomes determine a person's sex, whether the person is male or female. As in all mammals, human males have one X chromosome and one Y chromosome. This is called hemizygous. Females have two X chromosomes, and the female's X chromosomes are homologs. The remaining chromosomes found in both males and females are called autosomes. For both autosomes and sex chromosomes, we inherit one chromosome from our mother and one chromosome from our father. The life cycle of a multicellular organism is the sequence of stages leading from the adults of one generation to the adults of the next. Having two sets of chromosomes, one inherited from each parent, is a key factor in the life cycle of humans and other species that reproduce sexually. Humans, as well as most other animals and many plants, are said to be diploid organisms because all the body cells contain pairs of homologous chromosomes. The total number of chromosomes in humans is 46. This is the diploid number, and this is represented by a 2N. The exceptions are the egg and the sperm cells known as the gametes. Made by meiosis in an ovary or testis, each gamete has a single set of chromosomes, 22 autosomes, plus a sex chromosome, either X or Y. A cell with a single chromosome set is called a haploid cell. It has only one member of each homologous pair. For humans, the haploid number is 23. In the human life cycle, a haploid sperm from the father fuses with a haploid egg cell from the mother in a process called fertilization. The resulting fertilized egg, called a zygote, is a diploid. It has two sets of homologous chromosomes, one set from each parent. The life cycle is completed as a sexually mature adult develops from the zygote. Mitotic cell division ensures that all somatic cells of the human body receive a copy of all the zygote's 46 chromosomes. Thus, every one of the trillions of cells in your body can trace its ancestry back through mitotic divisions to a single zygote produced when your father's sperm and mother's egg fused about 9 months before you were born. All sexual life cycles involve an alternation of diploid and haploid stages. Producing haploid gametes by meiosis keeps the chromosome number from doubling in every generation. So let's move on to interphase. Interphase is the period of cell growth when the cell makes new molecules and organelles. Within the nucleus, the chromosomes are duplicated, but they cannot be distinguished individually because they are still in the form of loosely packed chromatin fibers. Most of the cell cycle is spent in interphase. This is a time when a cell performs its normal functions within the organism. For example, a cell in your stomach lining might make and release enzyme molecules that aid in digestion. During interphase, a cell roughly doubles everything in its cytoplasm. It increases its supply of proteins, increases the number of many of its organelles, such as mitochondria and ribosomes, and it grows in size. Typically, interphase lasts for at least 90% of the cell cycle. So from that standpoint of cell reproduction, the most important event of interphase is chromosome duplication, when the DNA in the nucleus is precisely doubled. This occurs approximately in the middle of interphase, and in the period when it's occurring is called the S phase. That stands for DNA synthesis. The S is for synthesis. The interphase periods before and after the S phase are called the G1 and G2 phases respectively. G stands for gap. During gap 2, or G2, each chromosome in the cell consists of 2 identical sister chromatids, and the cell is preparing to divide. And that brings us to meiosis 1. Meiosis is the process that produces haploid daughter cells in diploid organisms. It resembles mitosis, but it has 2 special features. The first is that the number of chromosomes is reduced to half. In meiosis, a cell that has duplicated its chromosomes undergoes 2 consecutive divisions called meiosis 1 and meiosis 2. Because one duplication of the chromosomes is followed by 2 divisions, each of the 4 daughter cells resulting from meiosis has a haploid set of chromosomes, only half as many chromosomes as the starting cell. The second special feature of meiosis is an exchange of genetic material, or pieces of chromosomes, between homologous chromosomes. This exchange, called crossing over, occurs during the first prophase of meiosis. As you go through meiosis, keep in mind the difference between homologous chromosomes and sister chromatids. The 2 chromosomes of a homologous pair are individual chromosomes that were inherited from different parents, one from the mother and one from the father. The members of a homologous pair of chromosomes are colored red and blue on your slide to remind you of this. In interphase, just before meiosis, each chromosome duplicates to form sister chromatids that remain together until anaphase of meiosis 2. Before crossing over occurs, sister chromatids are identical and carry the same version of all their genes. So, in prophase 1, as the chromosomes coil up, special proteins cause the homologous chromosomes to stick together in pairs. The resulting structure has 4 chromatids. With each set of chromatids, the homologous chromosomes exchange corresponding segments, or they quote-unquote cross over. Crossing over rearranges the genetic information. As prophase 1 continues, the chromosomes coil up further, a spindle forms, and the homologous pairs are moved towards the center of the cell. So this next slide just summarizes interphase and prophase 1. So as you can see, during interphase, the chromosomes are duplicated and the resulting sister chromatids are held together at the centromere. Then when we move into prophase 1, the chromosomes condense and the nuclear envelope fragments. Homologous chromosomes bind firmly together along their length, forming a tetrad. Chiasmata form between non-sister chromatids. Crossing over occurs at the chiasmata. Spindle fibers emerge from the centrosomes. Homologous chromosomes are attached to the microtubules of the spindle at the fused kinetochore shared by the sister chromatids. Chromosomes continue to condense and the nuclear envelope completely disappears. And that brings us to metaphase 1. At metaphase 1, the homologous pairs are aligned in the middle of the cell. The sister chromatids of each chromosome are still attached at their centromeres, where they are anchored to the spindle microtubules. Notice that for each chromosome pair, the spindle microtubules are attached to one homologous chromosome, come from one pole of the cell, and the microtubules attached to the other chromosome come from the opposite pole of the cell. With this arrangement, the homologous chromosomes are poised to move toward the opposite poles of the cell. So this slide is the continuation of the slide we were looking at 2 slides ago. And we see here we've added metaphase 1 to our table, where the homologous chromosomes randomly assemble at the metaphase plate, where they have been maneuvered into place by the microtubules. And this helps to create genetic diversity. Moving on to anaphase 1. In this phase, the attachment between the homologous chromosomes of each pair breaks, and the chromosomes now migrate towards the poles of the cell. In contrast to mitosis, the sister chromatids migrate as a pair instead of splitting up. They are not separated from each other, but from their homologous partners. In telophase 1, the chromosomes arrive at the poles of the cell. When they finish their journey, each pole has a haploid chromosome set. Although each chromosome is still in duplicate form, usually cytokinesis occurs along with telophase 1, and two haploid daughter cells are formed. And that brings us back to our table summarizing meiosis 1, and you can see now we've added telophase 1 and cytokinesis to our table. So just to summarize telophase 1 and cytokinesis, sister chromatids arrive at the poles of the cell and begin to decondense. A nuclear envelope forms around each nucleus and the cytoplasm is divided by a cleavage furrow. The result is two haploid cells. Each cell contains one duplicated copy of each homologous chromosome pair. And that brings us to meiosis 2. Meiosis 2 is essentially the same as mitosis. The important difference is that meiosis 2 starts with a haploid cell that has not undergone a chromosome duplication during the preceding interphase. During prophase 2, a spindle forms and moves the chromosome towards the middle of the cell. The nuclear envelope breaks down, the mitotic spindles bind to both sides of the centromeres, and condensation of chromosomes occurs. During metaphase 2, the chromosomes are aligned as they are in mitosis, with the microtubules attached to the sister chromatids of each chromosome coming from the opposite poles of the cell. And this slide gives us an overview of meiosis up to metaphase 2. The takeaway from this slide is the genetic arrangements. Notice we have 4 genetic arrangements on this slide. So you notice that in genetic arrangement 1, you have all blue chromosomes, and in genetic arrangement 2, you have all red chromosomes. But then when we look at genetic arrangement 3, we have a red and a blue chromosome in the cell, and same for genetic arrangement 4. So you can see how genetic diversity can start randomly arising through the process of meiosis. In anaphase 2, the centromeres of sister chromatids separate, and the sister chromatids of each pair move toward opposite poles of the cell. In telophase 2, nuclei form at the cell poles, and cytokinesis occurs at the same time. There are now 4 daughter cells, each with the haploid number of single chromosomes. This slide summarizes meiosis 2. We can see at prophase 2 the sister chromatids condense, a new spindle begins to form, and the nuclear envelope starts to fragment. The nuclear envelope disappears, and the spindle fibers engage the individual kinetochores on the sister chromatids. Then at metaphase 2, the sister chromatids line up at the metaphase plate. During anaphase 2, the sister chromatids are pulled apart by the shortening of the kinetochore microtubules. Non-kinetochore microtubules also lengthen the cell. In telophase 2 and cytokinesis, the chromosomes arrive at the poles of the cell and decondense. The nuclear envelopes surround the 4 nuclei. Cleavage furrows divide the 2 cells into 4 haploid cells. And in this slide, we see the process of meiosis summarized in one table. So you can see it starts with interphase, it goes to meiosis 1, and it goes to meiosis 2. Moving on to how meiosis helps create genetic diversity. As we discussed earlier, offspring that result from sexual reproduction are genetically different from their parents and from one another. When we get to evolution, we will see that the genetic variety in offspring is the raw material for natural selection. But for now, let's focus on meiosis and fertilization and see how genetic variety arises. So for starters, let's take a closer look at crossing over. Crossing over is the exchange of corresponding segments between chromatids of homologous chromosomes. This occurs during prophase 1 of meiosis. At the time crossing over begins, homologous chromosomes are closely paired along their lengths with a precise gene-by-gene alignment. The sites of crossing over appear as X-shaped regions, and this is called a chiasma, or plural chiasmata. The homologous chromatids remain attached to each other at the chiasmata until anaphase 1. The exchange of segments between non-sister chromatids, one maternal chromatid and one paternal chromatid of a homologous pair, adds to the genetic variety resulting from sexual reproduction. If there were no crossing over, meiosis could only produce two types of gametes. These would be the ones ending up with the parental types of chromosomes. Remember from our example on a previous slide how the maternal chromosomes were represented in red and the paternal ones were represented in blue? Well, if there were no crossing over, the gametes would be all red or all blue, or all inherited from your mother or all inherited from your father. With crossing over, gametes that arise have chromosomes that are part red and part blue, or partially inherited from your mother, partially inherited from your father. These chromosomes are called recombinant because they result from genetic recombination. Genetic recombination is the production of gene combinations different from those carried by the parental chromosomes. Because most chromosomes contain thousands of genes, a single crossover event can affect many genes. When we also consider that multiple crossovers can occur in each pair of homologous chromosomes, it's not surprising that gametes and the offspring that result from them can be so varied. Independent assortment is another way in which meiosis contributes to genetic variety. When aligned during metaphase 1, the side-by-side orientation of each homologous pair of chromosomes, consisting of two sister chromatids, is a matter of chance. Either the red or blue chromosome may be on the left or the right. Thus, in this example, there are two possible ways that the chromosome pairs can align during metaphase 1. In possibility 1, the chromosome pairs are oriented with both red chromosomes on the same side. In this case, each of the gametes produced at the end of meiosis 2 has only red or blue chromosomes. In another possibility, the chromosome pairs are oriented differently. In this possibility, there is a red chromosome and a blue chromosome on the left, and a blue chromosome and a red chromosome on the right. This arrangement produces gametes with one red and one blue chromosome. Thus, with the two possible arrangements of this example, the organism will produce gametes with 4 different combinations of chromosomes. For a species with more than 2 pairs of chromosomes, such as the human, every chromosome pair orients independently of all the others at metaphase 1. During meiosis, the X and the Y chromosome behave as a homologous pair, though they are not homologs. For any species, the total number of chromosome combinations that can appear in gametes is 2 to the n, where n is the haploid number. So where n equals 2, the number of chromosome combinations is 2 to the second power, or 4. For a human, where n equals 23, there are 2 to the 23rd, or about 8 million possible chromosome combinations. This means that every gamete a human produces contains about 8 million possible combinations of maternal and paternal chromosomes. How many possibilities are there when a gamete from one individual unites with a gamete from another individual during fertilization? A human egg cell representing about 8 million possibilities is fertilized at random by one sperm cell representing one of about 8 million other possibilities. By multiplying 8 million by 8 million, we find that a man and a woman can produce a diploid zygote with any of 64 trillion combinations of chromosomes. So we see that the random nature of fertilization adds a huge amount of potential variability to the offspring of sexual reproduction. And on this slide, we see a karyotype. This is a karyotype of a human female. How can you tell? Well, you can tell because at the sex chromosome, it is an X, not an X and a Y. If this karyotype had an X and a Y, this would be the karyotype of a male. So you can see for the human, as I've stated numerous times throughout this lecture, that there are 23 pairs of chromosomes. In this day of an age, most expecting parents will use a karyotype to identify any chromosomal anomalies in their expected child. A karyotype can also be used to determine what the gender of the child may be. Moving on, so far our discussion of meiosis has focused on the process as it normally and correctly occurs, but what happens when an error occurs in the process? Such a mistake can result in genetic abnormalities that range from mild to severe to fatal. Within the human body, meiosis occurs repeatedly as the testes and ovaries produce gametes. Almost always, the mitotic spindle distributes chromosomes to daughter cells without error, but occasionally there is a mishap called non-disjunction in which the members of a chromosome pair fail to separate at anaphase. Non-disjunction can occur during meiosis I or meiosis II. In either case, gametes with abnormal numbers of chromosomes are the result. When a normal sperm fertilizes an egg with an extra chromosome, the result is a zygote with a total of 2N plus 1 chromosomes. Mitosis then transmits the abnormality to all the embryonic cells. If the organism survives, it will have an abnormal karyotype and probably a syndrome or disorder caused by the abnormal number of genes. In the case of Down syndrome, the individual will have 3 number 21 chromosomes, making 47 chromosomes in total. This condition is called trisomy 21 or Down syndrome. In most cases, a human embryo with an atypical number of chromosomes develops so abnormally that it is spontaneously aborted or miscarried long before birth. However, some aberrations in chromosome number seem to upset the genetic balance less drastically, and individuals with such abnormalities can survive. These people usually have a characteristic set of symptoms called a syndrome. A person with trisomy 21, for instance, is said to have Down syndrome. This is named after John Langdon Down, who first described it in 1866. Affecting about 1 out of every 700 children, trisomy 21 is the most common chromosome number abnormality and the most common serious birth defect in the United States. Down syndrome includes characteristic facial features, frequently a fold of the skin at the inner corner of the eye, a round face, and a flattened nose, as well as a short stature, heart defects, and susceptibility to leukemia and Alzheimer's disease. People with Down syndrome usually have a lifespan shorter than normal. They also exhibit varying degrees of mental retardation. However, individuals with the syndrome may live to middle age or beyond, and many are socially adept and can function well within society. Non-disjunction in meiosis does not affect the autosomes. It also can affect the sex chromosomes. Unusual numbers of sex chromosomes seem to upset the genetic balance less than unusual numbers of autosomes. This may be because the Y chromosome is very small and carries relatively few genes. Furthermore, mammalian cells normally operate within only functioning X chromosome because other copies of the chromosome become inactivated in each cell. An extra X chromosome in a male, making him XXY, produces a condition called Klinefelter syndrome. Men with this disorder have male sex organs and normal intelligence, but the testes are abnormally small. The individual is sterile, and he often has breast enlargement and other feminine body contours. Klinefelter syndrome is also found in individuals with more than 3 sex chromosomes, such as XXYY, XXXY, or XXXXY. These abnormal numbers of sex chromosomes result from multiple non-disjunctions. Human males with a single extra Y chromosome, XYY, do not have any well-defined syndrome, although they tend to be taller than average. Females with an extra X chromosome, XXX, cannot be distinguished from XX females except by karyotype. Females who are lacking an X chromosome are designated XO. The O indicates the absence of a second sex chromosome. These women have something called Turner syndrome. They have a characteristic appearance, including short stature and often a web of skin extending between the neck and the shoulders. Women with Turner syndrome are sterile because their sex organs do not fully mature at adolescence, and they have poor development of breasts and other secondary sexual characteristics. However, they are usually of normal intelligence. The XO condition is the sole known case where having only 45 chromosomes is not fatal in humans. The sex chromosome abnormalities in which I've described to you here illustrate the crucial role of the Y chromosome in determining a person's sex. In general, a single Y chromosome is enough to produce maleness regardless of the number of X chromosomes. The absence of a Y chromosome results in femaleness. And this next slide illustrates 2 types of non-disjunction for you. At the top of the cartoon, we see non-disjunction occurring during meiosis 1, and in the bottom of the cartoon, we see non-disjunction occurring in meiosis 2. And this slide is a graph that shows the chance of having a baby with Down syndrome rises with the age of the mother. So before we adjourn, let's do a quick recap of tonight's lecture. First of all, you should know that there are 2 types of chromosomes in humans and what they are. You should be able to list the major layers of chromosome structure. You should know in which phase of interphase the sister chromatids form. You should know what the stages are of meiosis 1 and meiosis 2. You should know the unique events that happen during meiosis 1. You should be able to draw the stages of meiosis. You should know how and when genetic diversity is increased. You should know what is used to help diagnose chromosomal errors. And you should know what term is used to indicate an error happening during meiosis.